Good morning, everyone. We're going to get started with our presentations. So the first presentation of the day will be presented by Katie Durham, and her advisor is Dr. Jane Harris. She will be presenting on calculating maggot mass temperature in covered and uncovered human remains. So if you would welcome her to the podium with a round of applause.
Good morning, all. So my McNair research is on calculating maggot mass temperature in covered and uncovered human remains. Just a fair warning, this presentation does discuss human decomposition, and there will be images of parts of the body showing various states of decay. Once we get to this point in the presentation, I'll give you an advance notice and a heads up, and there will also be a symbol to let you know of any images on the next slide. So this is all the information we will be covering. To start off, my research is mostly based in forensic entomology, which started in the 13th century China. This is written in the Washing Away of Wrongs, in which it's the first incident actually accounted for. And the story is that someone is murdered in a rice field, and then an investigator comes in and is looking for who did this. So they take all of the rice workers and they line them up with their sickles, and one specific sickle has a bunch of flies attracted to it. And then later on, the person confesses to the crime that they committed, and these flies helped to identify this person as they sensed traces of blood that couldn't visually be seen, but the chemicals could to the flies. So this moves us to the 20th century, and now we know that certain chemicals and other things can modify insect behavior, and we can also use blowfly larvae or moth larvae and blowfly pupae to narrow down certain things in forensic entomology. We also use entomotoxicology and environments and species identification for this as well. These insects help us with postmortem interval, which is the time since death, so just trying to understand the time frame in which that occurred. And then the importance of the fly life cycle is very well known. Due to controlled laboratory experiments, it's very predictable, and it is also commonly found with decomposing organisms. So the fly life cycle at 70 degrees Fahrenheit is most predictable, and it takes 24 hours for larvae to hatch. And then this can range depending on the temperature. So if the temperature is higher, then they can hatch sooner. If it's colder, then they can hatch a lot slower. It just depends. And the fly life cycle starts as eggs, and then they become larvae. And then within the larva stage, there's 3 different stages, which is first instar, second instar, and third instar. And then they pupate, so they wander away from the decomposing organism, and then they turn into adults. And all of this fly activity is highly dependent on temperature and access. So what that means is if it was in a building somewhere and there was like no access inside, so there were like no windows open or anything like that, then chances are there's probably not going to be any fly activity there, and that's going to decrease their access. And along with clothing, certain clothing, or if they're in like a suitcase or any sort of thing in which outside can't get in, that would also affect this. Then we also have taphonomy, which is from the geological language. It describes anything that happens after death to an organism. And in forensic taphonomy, we are looking at a specific time frame in which we ask 2 questions. How long ago did this person die, and how did they come to be in their present condition? And there's many tools that can be used to assess for this, which is just a visual assessment, chemical, biological samples, and weathering, or skeletal, just looking at skeletal remains. So now we have accumulated degree days, and this is commonly used for relating time to temperature. And these are values that are calculated from from ambient temperature, or like using the National Weather Service data that's provided to calculate the time and the temperature. So for an example to better understand this concept is if you placed a body outside in Arizona in December compared to here in Marquette in December, the temperatures in Arizona would be a lot hotter and warmer, and the rate would be increased as well. Whereas if it was here, it'd be slower and it would have a lot longer time. So our research questions is to determine if maggot masses have a significant effect on temperature in uncovered and covered remains. The purpose of this study is to understand how maggot masses do affect temperature and how this is relevant to PMI, and if this could possibly change the equation we use to calculate PMI. Could this throw this off by a day and rule out a certain time, or does it have no effect at all? So the hypothesis for this is maggot masses have a significant increase of temperature when in an uncovered and covered condition of the donor compared to without any maggot masses. So the materials and methods. So the materials we used for this, we did use human remains, which are from the NMU Forensic Research Outdoor Station. All of these are donors. They're voluntarily donated to our program for research and educational purposes. The other materials we needed was a blue tarp for the covered donor, and then we used an infrared thermometer to check some temperatures, and these thermometers that you see in the corner here are called iButtons. There are wireless thermometers in which we could place in various locations to record maggot mass temperatures. And next to that is a capsule, and this is so that we could protect them because they could not get wet, otherwise the thermometer would break. So it's just a sealed capsule for those. And then we have the other photo is a picture of the Forensic Research Outdoor Station, also known as FROST. And here you can see in the bottom corner of the photo, there are these cages. These are used to cover our donors so that no other scavengers or animals can get them, so they're just protected that way. And then the donors we used for this, they were both males and approximately the same size, and they were both previously frozen. So our methods, we did decomposition data, so we took notes of the condition of each donor every day, and then we total body scored them, which is just assigning them a rate on how they looked each day, and it's split up into categories, so like the head, the torso, and the limbs. And then we took maggot samples, which we chose these by the areas of greatest concentration each day we went out. And then we took a spoon and scooped them up into a strainer, poured some boiling water on them, and then we would spray them with ethanol to rinse them off. And then we filled a little clear jar and labeled it with the date of the donor and the location in which they were taken from. And then they were stored in ethanol, and we did this study for a length of 31 days. And for the first 2 weeks, we went out every single day, and then after that, we reassessed, and then we went out every other day. And then for the statistical analysis of this, we used RStudio. So here is a diagram of where all the thermometers were placed for the maggot masses, which is the mouth. The uncovered one has one in the groin area, and then there's one on the right hip, and then one underneath, in the middle of the back. And then for the covered one, it's in the mouth area, the chest cavity, it's also under the back, and the right hip. All of these locations were chosen because they're most commonly known spots, and maggots most likely like soft tissue, so the mucous membranes. Okay, so now There will be human remains on the slide. The green star gives you a heads up. If you don't want to see any of these photos, please close your eyes, turn around, or leave, whatever you need to do. So this is data collection. This is just the process in which I was talking about previously. This is day 4, which is exciting because our study took off, and this is when the eggs really started to appear. This is day 7. You can see first instar maggots here as it's progressing throughout. This is day 10. Our study's only going up from here. These are our results. So this is just based on each location of every different eye button to the ambient temperature of the eye buttons, which was, we used a proxy, which was a surface temperature for the ambient temperature. And so basically our results showed that the area of the body that has the most variable is the oral cavity. The graphs demonstrate higher temperatures are located in maggot masses for sure. And the more frequency of higher temps, so maggot masses do increase the temperature. And then we're not really seeing any differences between uncovered and covered remains. The significance of this is that ambient temperature is what is generally used for forensic entomology. So if we focus more on maggot masses, then that could possibly alter our equation used for that. And then the significance is both covered and uncovered increase the local temperature. And the knowledge of these based on laboratory studies could affect how the calculation is made. And then limitations of this would just be availability and timing. And then some future directions of this would be to replicate this study at other taphonomic research sites, do it under various weather conditions, and just have multiple samples. And I just want to say thank you to McNair and Lindsay and Lee and my mentor, Dr. Harris, in the Fall Frost program for everything that they do.
We'd like to open the floor for a couple minutes of any questions that you may have, so feel free to raise your hand, ask your question.
Yes.
Where do you see this research going in the future? Like, what's your next step?
Ideally, I would like to kind of keep testing it and looking at it at different locations, because there are weather conditions in which maggots may not be there necessarily, so I don't know how relevant it would be to that. But for specific locations like around here, hopefully seeing how this does affect the PMI estimations, and maybe hopefully benefiting that, in which we can have more precise answers when it comes to crime scene response type of things and stuff. Yes?
So how did you become interested in this?
I actually wasn't really sure what I wanted to do. I knew I loved forensic anthropology stuff, and I was just reading a lot of stuff. And then I met Mandy, and she was talking to me a lot about it, and then I started getting into reading a lot of forensic entomology stuff, and I was like, actually, I think I want to do something with this. And I think temperature is something that I'm actually pretty interested in, so seeing how temperature is affected by other organisms was something I was curious about. Yeah. Yes?
So what was the most challenging and the most rewarding experience?
Oh, the most challenging was probably just never knowing what was gonna happen. It was a lot of waiting game, and it was kind of those wireless buttons we had. We basically started them on a computer and put them out in the field, and we had to wait and see if they would actually work and if they were collecting data all the way until the end of the study. So that was very nerve-wracking. But then it was collecting data the entire time. So I did have data by the end of the study, that was the most rewarding thing ever, that they actually worked. Because they made it through rain and all kinds of stuff. Yeah. I think we're good. Yeah. Thank you.
All right, I'd like to welcome up our next presenter, Jessica Laxo. She will be presenting on Play is Possible. And here's your, so let's give her a round of applause.
Go ahead.
And then slide advance if you want to use it. So right arrow key will advance to the next slide.
This one? Thank you. Hi, my name is Jessica Laxo, and I am a graduate student in the Early Childhood Education Program. I'm also a preschool kindergarten teacher up at Powell Township Schools, and I did a literature review for this project, and I investigated a problem that is vital for everyone, not just teachers, not just parents, and not just school administrators, but everyone. The future of our society are the children. And if we want a society that is creative, that is intrinsically motivated, curious, and can solve problems as a group, then we need to support developmentally appropriate practices in our public schools. And today I'm focusing on what the Kennedy Centers has deemed crucial. Forgot my first slide. There it is. It's a beautiful picture of me. But anyway, so I'm going to focus on what the Kennedy Centers has deemed crucial for our youngest learners. And that is that a good early education has lifelong benefits. And we need to support this by ensuring that the education for these young learners is developmentally appropriate. So I think it's really easy to say that play is important for early education. And sadly, Peter states that historians and social scientists have documented that play has been disappearing from the kindergarten classroom for the past 50 to 60 years. Piroletta et al. States that there is a global emphasis on early academics, and this could contribute to the decline of play in early education. The natural development of childhood is being replaced with desk time and listening to the teacher in the front of the room talk about subtraction, addition, what letters make what sounds, and how to properly write each one of those letters, and any other academic standard that kindergarten teachers are required to teach. And it is not within a 5-year-old's nature to sit still for long periods of time and memorize all these facts. But what if I told you that there is a way to meet all of those state standards and to do it with play? Well, sounds kind of magical, right? It is. And honestly, kindergarten should be full of magic, and it is full of wonder. So first, let's take a look at what's going on right now. When you look at a publicly funded classroom, there is a set of standards that must be met, and it's typically measured through standardized tests. Unfortunately, the research from the Center of Developing Child at Harvard University shows that the unintentional consequences of the pressure of these assessments and the continuation of adding more regulations for kindergarten is that there is a rise of teacher-directed activities. And this means longer periods of time sitting, which means a lot less movement and less peer-to-peer interactions. And according to Zosh et al., when there is less play in a classroom, healthy development and learning is actually being hampered. So let's take a look at that magical classroom that I spoke of earlier. When you hear, or when most people, maybe not you, hopefully not you, but when most people hear the phrase a play-based classroom, an image of a group of 5-year-olds running and screaming through a classroom is really easy to imagine. So thankfully, Zosh et al. Has defined several types of play. There's free play, and there's games, and there's guided play. So free play is what most people think about. It is the kind of play that allows the children the freedom to create their own play rules. And this is crucial. This is really important for kindergartners. Piroletta et al. Ensures us there's a lot of social learning that happens during that free play. And games, well, games are just that. They can be the board games. They can be games that either children or teachers create the rules, or have the rules and they can lead them. And guided play. Now, guided play is what I want to focus on today, and that is what is the magic in a classroom. So 5-year-olds, they want to explore, and they want to learn. And they are at the most beautiful age that is so full of curiosity. And a teacher can harness that curiosity and guide their learners through play to expand their knowledge. So you might be wondering, how can you possibly teach literacy through play? Well, for example, in my art area during free choice, I frequently witness early literacy skills as my students create their own stories, whether with words or with pictures, and they bring them home so they can read them to their parents or to their siblings. And one of my favorite ways in my classroom to combine play with reading and writing is allowing the students to help with the dramatic play area. I have a couple examples up there on the far right. And in my classroom, we switch over our dramatic play area every month. And the kindergartners, they are in charge of creating all the signs, all the menus, or any other written information that goes into that area. And this helps create ownership in that classroom while they get to practice that letter formation and those letter sounds. Well, what about math? How can a teacher possibly teach math through play? Well, an example that Zosh et al. Talks about is to play Shape Detective. So once all the students are gathered, put on your best detective outfit, you know, the cap, grab a magnifying glass, go to Goodwill, get a cape, play it up. And you're gonna start with whatever shape you want them to learn about. For example, if you want them to learn about triangle attributes, hand each student a different triangle: equilateral, obtuse, acute, right angle. And then in your best detective voice, you're gonna ask that class what they notice about the shapes they have. How are they all similar? And then you're gonna start to add in that vocabulary. Yeah, all those shapes have 3 sides. They're all triangles. Triangles have 3 sides. Now this can be done with every single shape: hexagon, octagon, trapezoid. It's a lot of fun. So number recognition, that can also be practiced through play. And one of the games I love to play with my class is called Musical Hoops. And this is truly a favorite game amongst my students. And what I do is I'm gonna take hula hoops, or you can draw large circles on the ground. And in each one of those circles, you're gonna put a number. And then you're gonna start playing the music. And you're gonna tell the students, when you hear that music stop, I'm gonna yell out a number, and you're gonna find that number, and you're gonna stand in that hoop. So what about social emotional learning? Teaching children how to handle their emotions and how to respond to their peers' emotions are now on an educator's shoulders, according to the Michigan Department of Education. So my classroom, we like to play Guess That Feeling. And what you do is you're gonna have students pair off, and you're gonna give one child an emotion, and the other child has to guess it, and then they switch. So we go through many different feelings, and my students, they thoroughly enjoy making faces at each other without me saying, why are you making that face at that person? So they really enjoy that. And they request that really at least weekly. And according to the Center on the Developing Child at Harvard University, we are currently dealing with an unprecedented amount of early childhood trauma. And the phrase from Laura Magass, when we play, we can withstand incredible hardships, is a phrase that we need to keep in the forefront of our mind. Because children cannot learn until they feel safe and connected. And through play, we can meet that. We can cultivate a classroom community that supports each child's development. So these examples I talked about are truly just scratching the surface of how to bring hands-on interactive play back into the classroom. The standards for kindergarten can still be met while allowing the children to have these enjoyable experiences. So now I have a job for you. And this is really important. So everybody actually needs to pay attention to this part. So I want you to go out into the community. And I want you to advocate for play-based education and our earliest learners. I want you to go out and I want you to support your early elementary school teachers for your community. And I want you to write those letters to the government supporting a play-based education so we can have inquisitive, intrinsically motivated, and lifelong learners that will eventually become the leaders of our community. And if you want to learn more, I highly recommend watching Gary Peters' TEDx talk called The Decline of Play. Or if you want to learn along with your child, listen to the science-based podcast, Mom in the World, episode 101, called Recess. Thanks. Oh. There you go.
If anyone has any questions they'd like to ask, feel free to now.
Yeah.
It's a little beyond the scope of your study, but do you feel that there's room for playfulness in higher education?
Yes, 100% yes. So I talk a lot about this. I have ADHD, and until I became a graduate student, I didn't realize like truly how fun learning could be. And it's because I get to add in, I get to work with kids. I mean, really, that's a lot of fun. And I get to add in that play for myself. Yeah, so yes, I think there is.
Be happy to collaborate with you on a study of that.
Sure. If you can handle my my moving, I like to move a lot, so if you couldn't tell. So
I'm a doctor after.
okay. Yeah.
I'm curious if in your review of the literature, if you came across anything relating. So one of the last things you said made me think of self-determination theory, like being intrinsically motivated with autonomy and
Yeah.
that lifelong learning piece. If you came across any literature where folks were kind of using that framework in the context of this early childhood education.
Yeah, so a lot of literature, a lot of current literature really talks about having that self-autonomy for kids to learn. And I think that's, I feel like there's a lot of gaps right now because you have this literature that says, yes, play-based learning is really important. This is what we need. And then you have these state standards and you have the government saying, well, we need to assess this and we're going to do this through standardized tests. And here are all these standards. And how are you meeting this? And so while over here in this bright rainbow glitter-filled plane that these kids get to choose and are student-led, so they are motivated, so they can create all of those neurons that happen during the early childhood. Over here you have people saying, well, that's great, but how can you show us that? And I think bridging that gap between the two, there's not much of a bridge there.
So it sounds like there's a gap.
Yeah.
Thank you.
Welcome. Anybody else? Awesome. Thank you.
Marissa?
Yeah.
Perfect.
I spilled my water all over myself.
And I'll be sitting there, and I'll have a time warning at 1 minute 30 seconds, and then a stop.
Is it okay if I also?
Of
do
course,
this?
go ahead. I won't start it until I get back to my seat.
Thank
And
you.
then
So
this
is
is
it
a
right when you
slide
get back to us,
advancer. If you want to use it, you can also just use the arrow key.
Okay, I'm just confused.
Perfect.
There's too many things to learn right now.
All right, folks, I'd like to welcome up our next presenter, Marissa Berkman.
Yeah.
Thank you. And she'll be presenting on predictors of NMU student success in STEM courses related to campus-provided resources. So please join me in welcoming her.
Thank you, guys. All right. Good morning, everybody. My name is Marissa, and I'm an undergraduate student in biology at Northern Michigan University, and I'm presenting on tutoring-centered usage data as a means of analyzing predictors of NMU student success in STEM courses related to campus-provided resources here at NMU. So peer-led teaching has been shown to improve learning outcomes for students of all ages. One study correlated high use of tutoring centers, defined as 10 or more visits per semester on campus, as coming out with GPA grades as higher than 0.2 as compared to non-users. Our tutoring center on campus, All Campus Tutoring, is free for all students and covers around 40 subjects. Oh my gosh. Although most hours of operation are between 6 and 9 p.m., and aside from nursing classes, most courses covered fall between the 200 level and below. I went to 2 universities before coming to Northern, and I also worked for All Campus Tutoring as a tutor. I noticed that this tutoring center is normally a lot less full than my last 2 universities' tutoring centers, and I wondered what are some academic predictors of tutoring center usage, if any, on NMU's campus. We chose to ask about self-reported GPA and mathematics grades because both are documented predictors of student success in STEM courses, allowing us to assess the relationship between achievement level and tutoring center usage. College history, including semesters in college, transfer history, and intended major, was used to determine risk groups that may benefit from simple interventions like better advertisement of campus resources. All Campus Tutoring self-reported usage was used to determine the location, frequency, and content that tutoring was used for. Finally, high school and education zip code was gathered in lieu of parents' educational and financial background to determine median hometown income and educational background as a means of protecting anonymity of participants. Our main priority was to keep information completely anonymous during this initial dive. We hypothesized that tutoring center usage would primarily consist of high-achieving students, as indicated by an NMU GPA greater than 3.0. The GPA 3.0 was chosen as a somewhat arbitrary definition of high-achieving student, considering that a 3.0 student would get, on average, B's in their classes. This was based on the assumption that high-achieving students either actively sought out tutoring and other academic enhancing opportunities, or that students who used these opportunities actively benefited from them. We also hypothesized that most tutoring center usage would be composed of students having 4 or fewer semesters in college, given that the main tutoring centers are located in or near the dorms on campus, where students are required to live during their freshman and sophomore years. We used 2 methods to assess our hypotheses. Our voluntary online survey collected real-time data using fill-in-the-blank and multiple-choice questions. This survey was sent out with a mass email by Dr. Mormon on April 14th, and data was pulled on April 18th for our first analysis. We also used tutoring center data from fall of 2022 and historic grades from 2013 onward for biology and chemistry courses to draw conclusions regarding tutoring center need. All data was de-identified and preserved anonymity for all participants. This is the PowerPoint slide advertisement that was sent out to professors before we ran our survey to maximize response rate. And here are the results from our fall 2022 tutoring center and grade history analysis. This is a distribution histogram of grade count since 2013. Of important note, we can pull from this data that the relative number of failing and passing grades from each course and determine where tutoring center need lies. Since 2013, the highest enrollment courses, Bio 111 and 112, respectively, Principles of Biology and Biological Diversity, Chemistry 111 and 112, Gen Chem 1 and 2, and Anatomy and Physiology have enrolled and filled out the highest number of students and the highest rate of students of the 14 courses surveyed. Tutoring center analysis of chemistry and biology courses showed 516 visits total for the analyzed 14 courses, many of which are required for biology students. Interestingly, chemistry courses in general made up 91% of visits surveyed, and general chemistry 1 and 2 made up around 50% of visits surveyed. Comparing tutoring center usage to class enrollment in 2022 shows a huge discrepancy. We see a fairly homogeneous distribution of class enrollment on the right, with chemistry courses composing 41% of enrollment and biology courses composing 59% of enrollment in courses analyzed. We can see from these pie charts that tutoring center enrollment does not trend with class enrollment, with chemistry courses in general taking up a much larger proportion of tutoring center usage than enrollment. This led us to examine failing grades in these courses to determine where tutoring center resources may be best allocated. And as you can see, tutoring center usage also does not significantly correlate to classes that have historically failed out the highest number of students. Although Gen Chem 1 and 2 did have relatively high tutoring center usage in 2022, Bio 111, 112, and 207 have significantly lower tutoring center usage rates than total failing percentages of courses evaluated. Of special note, despite making up more than a quarter of failing grades since 2013 in the courses surveyed, Bio 111, Principles of Biology, made up only 7% of tutoring center usage of courses analyzed in fall 2022. Similar trends can be seen for D grades. The results of our voluntary survey somewhat paralleled the results from our data mining experiment, but gave us ideas on where to take this survey next. 41 students responded before our first data poll, averaging around a 10 student per day response rate. 54% of students reported never using the tutoring center, and we see a fairly consistent negative correlation between percentage of respondents and frequency of use at the tutoring center. We analyzed GPA as a predictor of tutoring center usage and found the results to be significant with a simple contingency test. We can see from the histogram of respondents comparing GPA and use that only respondents with a 2.1 GPA and higher responded to our survey. The most frequent tutoring center usage came from students with GPAs ranging between 2.1 and 3.5, and the majority of students with GPAs ranging from 3.1 to 4.0 reported going once or twice a semester to never. So this did not support our hypothesis that high-achieving students, as categorized by having a 3.0 or higher, sought out tutoring at higher rates than other students. We did, however, find that tutoring center usage was significantly linked to number of semesters in college, with highest representation coming from third semester college students and dropping off significantly after this point. Qualitative data analysis of free response questions showed that a number of students were unaware of courses covered by the tutoring center. Upper-level biology courses, particularly those heavy in terminology, were requested to be covered by a number of students. No students requested additional chemistry coverage, although a number did request biology courses that either are or are not already offered. As expected, chemistry courses were reported as the main content sought at the tutoring center by users. This study should be continued by using a dredge function to determine specific predictors of tutoring center usage specific to academic variables. Replications of this study should include ethnicity and other sociodemographic variables, given that NMU is composed of 87.5% white students. Additional future research may also include intervention implementation and grade distribution outcomes measured against tutoring center usage. Some short-term goals to increase tutoring center usage may be simple, like increased advertisement by professors, considering that some students don't know that needed subjects are offered. Bio 111, Principles of Biology, is a recommended need-based group based on its relatively high course enrollment, fail count, and fail rate compared to relatively low number of tutoring center visits. Biology courses in general seem to be underrepresented in tutoring center usage when compared to enrollment, fail rates, and self-reported need, and are suggested as a larger target group for future intervention. Resource distribution studies may show us how the tutoring center and other free resources on campus are used, and how they may be revised to increase efficacy in regard to student academic performance. I'd like to thank my faculty advisor, Dr. Mormon, for being an amazing professor, advisor, and mother to her young daughter all at once, Michelle Stephenson from All Campus Tutoring for providing our resources, and my own parents, Rick and Peggy Berkman, for assisting me in this project. Now I'd like to open the floor to any questions. Yeah.
Do you happen to track at all, like, a reattempt? You know, people fail the classes, then they take it a second, sometimes a third time. Like, do you know if there's any correlation there with usage of the tutoring?
No, we didn't check in on that, but we'd actually love to continue this study. I actually am starting grad school here this fall, so we are hoping to continue this study over the next couple of years.
Right.
So yeah, that's a good idea. We would love to include that. What other questions? Yeah.
I guess is the tutoring center in tandem with the instructors that are seeing that a student is actually struggling or failing grade-wise from the grade book? Does anything go out from the instructor to the tutoring center and or resources to the student that they see maybe struggling?
I think Dr. Mormon might be a little better equipped to answer this question, but I think it's really professor-based. I don't think there's necessarily...
Well, then obviously involved with the student as well.
Right, so most instructors
Yes, I would say yes.
So depending on the subject, you struggle to get students that are willing to tutor that subject. So that's another area that
Yeah.
we looked at, how can we incentivize it, or how can we kind of motivate those peers to come teach them.
I would say yes, we have like enough on some courses, definitely not enough in other areas. There's a lot of nights where we're working where we are just not doing anything. I think I've had maybe 5 visits all semester from people that I've actually had to help as a tutor. But then generally people who are teaching chemistry don't ever have free time. So if you're a chemistry tutor, you're using 100% of your time there as a tutor. Me as a physics tutor, I use most of my time there to do homework. So I think it's really class-based. Is there any other questions that I can address? Yeah?
So you identified that there was maybe some demand for the upper level biology courses.
Yeah.
I teach upper level chemistry.
Yeah.
I think the reason why you don't have tutors for these, or maybe the students are graduating.
Yeah, that's a big one. We'd almost have to employ grad students, and that's difficult on this campus because our TAs generally meet their max hours on campus that they can work doing their TA job. So even though grad students can work for the tutoring center, most of them choose not to, because why would you when you could become a TA and get your things paid for? Yeah, so it's kind of a catch-22. It's a difficult situation that we're in. Yeah, are there any other questions that I can address? Well, thank you guys for letting me speak today.
We have a couple minutes before the next one is going to start, so if anyone wants to jump up and grab some extra snacks before we start at 10:45, feel free. There's plenty of snacks. I have your presentation, Nathan. Keep getting your food, everyone, but I'm going to introduce our next presenter now. So our next presenter is Nathan Joyle, and he will be presenting on a study of the relative commercial viability of cannabis using organic and sustainable farming techniques. So if you would join me in welcoming Nathan.
Howdy. Well, first of all, where's Janelle at? I want to apologize to Janelle. Oh, she left for a minute. Yeah, I made her say mycorrhizal fungus like so many times. And one of the things I learned at 3-minute thesis was that I should have a more approachable title that just better conveys the point of my research. So, yeah, this is what I ended up going with. Yeah, so I think that the first, the most broadest question I can answer about my research is, why am I doing it? Why do we care about this research? And the simple answer to that is that salt fertilizers are bad. They are very much not good. They tend to build up in the soil, and they cause your farmland to become less fertile over time. And that requires more and more chemical treatment of your farmland. It leads to genetically modified crops to handle that. And another harmful side effect of fertilizers is they tend to run out of your farmland and get into water tables, where they cause algae blooms that displace the natural wildlife there, and can ultimately poison our groundwater supplies and our municipal water supplies, which causes us increased cost burden at that point to provide ourselves clean water. And all of that is really bad. Our world faces a record number of food crises in countries, and we are very privileged here in America. We don't see that a lot, but there are lots of countries where they literally cannot get enough to eat, and people die for that reason. So protecting our farmland is an important thing, and doing sustainable farming, I think, is the way that we're going to do that. The next question I get asked a lot is, why am I using cannabis to do this study? Why am I not using corn or soybeans or something? And I don't love that the low-hanging fruit or the very superficial answer is the answer here, but it is. The cannabis industry is huge right now. It's really growing, and there's a lot of corporations that have rapidly increasing budgets and rapidly increasing competition. And so there's a lot of potential for corporate sponsorship of research. And the lessons learned in cannabis can just as easily be applied to other crops, the same as we would with corn or soybean or anything else we studied. The other question I get asked a lot, because I'm studying cannabis, why is cannabis important? Why is this industry so huge? Why is it blowing up like this? And the answer that I have learned that I think is the most important is because each of us, every one of us in this room and watching at home, has within their bodies a system called an endocannabinoid system. And this system interacts with the over 113 cannabinoids that have been identified to naturally grow in the cannabis plant. And this endocannabinoid system, through the CB1 receptors in your nervous system and the CB2 receptors on your immune cells, touches almost every system in your body and provides regulatory control for all of these systems in a way where these chemicals can be incredibly beneficial medicinally. And I think you're only going to see more and more research focusing on this aspect of the cannabis plant in the future. Another question worth answering is, what do we mean by sustainable farming? That is maybe in some context a marketing term or a catchphrase. So to talk specifically about what we're doing on this project, sustainable farming in this case involves a process where we take an organic medium that is harvested from nature. In our project, we'll be using peat moss, but cocoa coir is another popular alternative. And we fertilize that with elements that are found only in nature, and often these things are waste products from other industries like bat guano or oyster shell. And this process is highly sustainable because of its minimal impact on the environment and the low cost of maintenance to it. It is a known process in many commercial industries, but it's typically viewed as a niche area of work because it has a perceived high cost to it. The upfront cost is very high relative to just dumping fertilizer on the ground. Talking a little bit more about the sustainable soil. Commercially, this process is known as super soil or a living soil, and that's due to the fact that it's populated with beneficial bacteria and fungus that harmonize with the plant, called mycorrhizal fungus. And those fungus and bacteria work symbiotically with the plants in the soil. And the soil can be regenerated, it can be recycled in a process called regenerative soil. And most of the products used to make it are OMRI listed organic. So the specific goals for doing this research for me and I think for my project team are to promote NMU as a Center for Sustainability Research and to demonstrate ultimately that the sustainable farming yields a better rate of return. That in spite of this perceived upfront cost, ultimately it is the more commercially effective option. And I think that proving that is the only way you're going to get corporations to adopt this kind of a process wholesale. And so then by using the cannabis plant, we can make implications about commercial agriculture in general. And ultimately, because of that eye on corporate sponsorship and that eye on changing the minds of corporations, the final answer I aim to generate is going to be in terms of return on investment. The neat thing about this project for me that I enjoy, I get access to this controlled environment agricultural space. It's this really neat setup that is totally indoors. It uses artificial light, and it's got humidity controllers and all this neat stuff. And it keeps the temperature for this nice range that the plants really enjoy and they really thrive in. It keeps the humidity right in this nice target zone we're looking for. And it has these two spaces. And you might ask, why do we have two spaces? And that is taking advantage of another interesting feature of the cannabis plant is it's highly photosensitive, which means it responds to how much light it receives. So the cannabis plant, like many fruiting plants, is looking for a certain window in the season where it likes to produce its fruit. And for this plant, that is 12 hours of light and 12 hours of darkness. And so by manipulating this, we can control when the plant generates fruit and when our harvest time is. So we can do these nice regular seasonal harvests repeatedly, independent of what the conditions or the seasons are outside. So the experimental plan for this will be to grow 8 crops, each of 9 plants. These plants will be identical clones, genetically identical copies of the same plant. And we will do 3 plants each times 3 different treatments, looking at, first of all, this organic, sustainable soil. And then we'll be looking at just topsoil with fertilizers dumped into it. And then we'll also be looking at a hydroponic system using fertilizers, which is common of indoor farming today. We'll collect one set of data biweekly looking at how big the plants are getting, whether or not they're showing any disease symptoms. And then we'll collect a second set of data at the end looking at what was the yield of these plants. Specifically, these measurements, we'll be looking at the height of the plants. We'll be looking at the content of the solution that the plants have access to, the nitrogen, potassium, and phosphorus that the plants are able to access. And we'll be making qualitative observations about disease conditions and such that run through the plants. We'll also be taking the dry weight of numerous aspects of the plants after harvest. This will lead to a lot of interesting comparisons. First of all, our primary goal is to determine the yield to cost ratio for these different methodologies and ideally show that the organic and sustainable method of farming yields a higher rate of return. We'll also be able to look at a number of aspects such as the access to nutrients as compared to the yield, the ratio of roots to stems to fruit, and look at how does the volume of roots the plants develop affect their ability to produce fruit. Lots of interesting questions like that. We'll put all this data into a mathematical model. I use a program called RStudio that does all the heavy lifting here. I'm not myself a super stats guy. It's a lot of hard work. I have a great advisor that is. But that's what I like about RStudio. You give it your data and it does all the thinking part for you. I'll be using a linear mixed model and I'll ultimately trying to derive yield as a function of treatment. I'd like to take a second before I move on to thank a couple of people. First of all, the fine folks at the College of Graduate Studies and Research. You guys do so much work for this event and other things. Thank you. And then my awesome advisory team. I see that some of them are here today. Dr. Leslie Putman, Dr. Donna Mackey, and Dr. Matthew Van Grinsven. You guys are just awesome. You've been so supportive, and I really thank you so much. And then in the interest of good ethics, I have this whole list of references here that I have. Nobody does anything alone. You build your work standing on the shoulders of people that came before you. Is there any questions? Right here for a second.
Which specific mycorrhizal fungus are you employing in this study?
That is a great question. I don't have that list on my person right now, nor do I have it memorized. I have a poster over there. I'll be hanging around there a lot of the day, and I can look that information up and give you a complete list. Ultimately, it's a commercial product I buy that's intended for soil amendment.
You had me at mycorrhizal fungus. We're staying for this.
Nice. Awesome. And then, over here.
I guess I was looking at it from the standpoint of, like you said, there's obviously more funding going into, allocating resources to this. And, the cost of being natural versus just dumping fertilizer on, or you've already told us that the longevity of that is going to be less fertile soil. If you're looking at a corporation that wants it right now, the yield, how do you combat that?
Well, I think that by ultimately breaking the process down into what is the cost per plant, and then evaluating yield metrics about what is the yield here, and developing then a yield per dollar spent, I think when we reduce it to that level, that the sustainable farming will show that it is a more profitable endeavor overall. And I'd like to believe that that math would be enough to at least open the door.
Because I've seen that model just in organic farming where yes, there's a better end product full of more nutrients. However, the corporations are looking for
Totally, yeah, and that's reducing it to a dollars kind of question is how I hope to combat that kind of an attitude. You got to make it matter to them. Yeah.
So, for the sustainable agriculture, I know with some fungi businesses, they use their waste product to provide nutrition to fields, and then another big tactic is leaving residues in the field, right? The products you want from cannabis don't have or a lot of nitrogen, phosphorus, and potassium in them, right? You could leave that in the field. The problem is transporting this. So, how big of a concern is going to be petroleum prices in your analysis, like the fluctuations in petroleum prices changing the answer you get?
That's a really great question. I honestly, I'm not considering transportation in my cost model at this point. I'm going to assume that you have all of your equipment on site, whether that's hydroponic equipment or your soil amendments. That would be a really great question to explore as a follow-up. And, over time, that's going to change too. If we end up looking at electrifying fleet vehicles and things like that, that would change that kind of a question. Obviously soil amendments and also liquid fertilizer are both heavy things that need to get trucked around on semi-trucks, and that's a huge impact to the environment, however you cut the cake. I do think the fertilizers need to be trucked in every time, all the time, whereas the field, like you're saying, is when you get your field to a certain point, you can minimize the amount of amendment you need. So I've done absolutely no analysis on that question, but from the hip, I'd like to think that the math might suggest that you have, over time, a less need to import material with the sustainable farming, but I have no idea.
What are your yield metrics? Is it biomass? Is it a specific secondary metabolite?
We are looking at the dry weight of the usable fruit, and then we're doing chemical analysis on that, looking at several secondary metabolites, yeah. Okay, if there's no more questions, I have a poster on display out here, and I'd like to thank the GSNR folks for squeezing my poster in at the last minute and making space for me. You guys are amazing. I will be standing around my poster off and on throughout the afternoon. If you think any more questions, I love talking about my work. I'll be there. And Janelle, you were gone. I have changed my title a little bit. I wanted to apologize to you for the number of times I made you say mycorrhizal fungus. I no longer use that in my title. I talk about sustainable farming now. Okay. All right. Thank you guys so much.
If you'd like to join me in welcoming our last presentation for this morning block, Sydney Crome will be presenting on biomass and carbon sequestration in the Dead River Community Forest. So join me in welcoming Sydney up here.
Okay, perfect, thank you. Alrighty. Hi there, everybody. My name is Sydney Crome, and I am a student in the Department of Biology here at Northern. So, as part of my participation in the Freshman Fellows Program, I conducted research on biomass and carbon sequestration in the Dead River Community Forest. So first, some background information. So what is biomass? So biomass is a measure of the mass of an organism, and it's typically used to compare species within and across ecosystems. But for our purposes, biomass is used as an indicator of carbon storage potential or carbon sequestration potential in trees. So the assumption here would be that as the biomass of the tree increases, so does its potential for carbon sequestration. So, as temperatures continue to rise, increased concentrations of carbon dioxide can be found in the atmosphere. And this exacerbates issues such as climate change and ocean acidification. So a healthier pathway for this excess CO2 to go is into trees and other vegetation through photosynthesis. So the trees would take in this CO2 during photosynthesis, and then they would convert it to biomass, so like the trunk, the foliage, the root system. So that would make them good fighters for climate change. But it is important to note that the ability to store carbon varies by species of tree. So I had the opportunity to partner with the Upper Peninsula Land Conservancy, or the UPLC. So the UPLC is a nonprofit organization based in Marquette, Michigan, and they manage plots of land throughout the Upper Peninsula with the goals of promoting recreation, education, conservation, and climate change mitigation. So in October of 2022, the UPLC acquired the Dead River Forest Community, the Dead River Community Forest, which I'll be referring to as the Dead River going forward. And then with their goals in mind, my research aims to address their climate change mitigation strategy for the area. So now we are going to take a journey. So starting here in James Rich Hall, we are going to head west along Highway 41. And then eventually we are going to hang a right and we'll end up at the Dead River Forest. So this highlighted portion right here is the Bayous parcel, and this is where I conducted my research. And then in a second here, it is going to zoom in on 4 lines. And these are my sampling transects, where I sampled my trees from. So these are some of the field tools that I used. We have a 100-meter transect tape, we have a diameter tape, a GPS, and then we used a camera to take canopy shots as we moved along. So what did I do in the field? Well, I used the point-centered quarter method. So if this was the Dead River Forest, for each transect at 20-meter intervals, I would establish a center point. And from the center point, I would extrapolate an imaginary perpendicular line so that 4 quadrants were created. And then in each quadrant, I would sample the 2 trees closest to the transect line, and I would record their diameter and their species. And here's me taking the diameter of a tree. Look at that smiling face. All right, so this data allowed me to generate estimates for total above-ground biomass using species-specific allometric equations. So in terms of ecology, allometric equations are when you take simple measurements such as diameter or height, and then you plug them into an equation that helps you calculate more complex measurements. So in this case, that would be biomass. So these equations are formulated by other researchers. So they would have physically gone out in the field and say cut down an eastern white pine. So they would record the diameter or height of the specimen, drag it back to their lab, dry it in a huge oven, and then record the biomass of it, or like the mass. And so they would compare their previous diameter measurements. So they would generate like a scatter plot with the diameter and the biomass, and then from there create a line of best fit. So when I record the diameter, I just plug it into the equation made by them, and then it presents me with the biomass of the tree. So results. So if you've never seen one of these before, this is a rank abundance curve. So rank abundance curves take into account species richness, which is the number of individuals, as well as species evenness, which is a comparison of species abundance between other species. So high species richness and high species evenness are indicative of optimal ecosystem health and function. And so visually on a graph, this would appear as a straight line or like a gently sloping line, which obviously is not the case for this data set. So the steep line right at the beginning there indicates that the majority of the biomass in the Dead River Forest is concentrated into 2 species,
those two species being the sugar maple and the eastern white pine. Okay, so I want everyone to imagine that there is an elephant in the room with us right now. So the average elephant has a biomass of 6.8 metric tons. So I sampled 297 trees, and from that, I estimated the total above ground biomass for my transects to be 283 metric tons. That is the equivalent of 42 elephants. So to take that a step further, sugar maples would have accounted for 18 elephants, where eastern white pines would be worth 8 elephants. So that's already a difference of 10 elephants worth of biomass. But if you take into account that there's not only sugar maples, there's also many other species of maples. The maple family in general accounted for 25 elephants worth of biomass, which is over half the biomass I estimated for the forest. So what does that mean? Well, okay. So eastern white pines tend to have a longer lifespan. They can live a very long time. And then in general, they are just a more massive species, so they're going to have thicker trunks, they're going to have more foliage, and a more extensive root system. So this makes them a great long-term candidate for carbon storage and climate change fighting. But then on the other hand, you have maple trees. So maple trees are a fast-growing species, so this means that they'll be sequestering carbon from the atmosphere at a faster rate. And then, at least in the Dead River Forest, they were significantly more abundant than the eastern white pine. So through sheer volume alone, they would be sequestering more carbon. So the UPLC will take this data, and they will weigh the pros and cons of the ecosystem services provided by both species, and then they will use that to inform their land management and climate change mitigation strategy for the area. So my research is just a preliminary assessment of the biomass distribution and composition of the Dead River Community Forest. My hope is that this will open the door for future research, such as into soil acidity and water table level for the forest. So soils with lower pHs or higher acidity tend to be lacking vital nutrients for trees. So some tree species are more tolerant of this nutrient fluctuation. And then on the other hand, with the water table level, ecosystems near rivers, such as the Dead River, tend to have higher water table levels, which isn't necessarily a bad thing, but some tree species are more tolerant to saturated soils than others. So this could also better inform the UPLC's land management strategy for the area. And then I would just like to take a second to acknowledge the freshman fellow staff and then Dr. Adam Naito, who helped me with my research. There we go. And then I would like to open the floor to any questions. Yeah. Definitely. So eastern white pines are huge trees. So they're less abundant in the area. So when you see an eastern white pine, it kind of draws your eye because it has a significantly bigger trunk. Yeah, and it's significantly taller. And then for sampling, we had a minimum diameter that the tree had to have for us to consider it, because some of them are just so young that they're not going to be sequestering much carbon. But the majority of the maples that we saw were actually young growth. So it'll be interesting to see in the future the potential there. Did you have another question? That, yeah. Well, the Dead River in general is a protected area. It was previously, I think it was privately owned previously, or owned by someone else that was not the UPLC. So they don't actually have much development in the area. But we did notice there was an old tire at one point. So people have definitely been there, but the land actually is not developed at all. Were there any other questions? Yes.
You described that your eastern pines were not very frequent, but they were very large, and that was why they sequestered a lot of had a lot of biomass. Comparatively, were the sugar maples just a lot more common, and that was
Yeah, they were significantly more abundant. That's actually a very interesting point because so we did the calculations and then we like summed them for the biomass, so it was like a calculation per species. But with the eastern white pine, like we only saw, I don't want to give like a number because I don't remember off the top of my head, but like including like the saplings, you would see hundreds of maple variations, but then you would only see a few eastern white pines. So the fact that they would even show up on such a high level for the biomass distribution is like a huge thing altogether. So yeah. Yes?
So my question's about the model that you use from other researchers. Are the error bars on those estimates the same across tree species, or are they different?
I don't know about the error bars. As to the only thing I can really speak for to the equation is that it estimates above ground biomass, and that they were extensively researched and like drawn from scientific literature. But I don't. We were provided with numbers, so not so much error bars, if that makes sense. Any other questions? All right, thank you.
All right. So right now we're going to take a break from our oral presentation. So feel free to grab snacks, wander around, and check out the many, many posters we have out there. And we'll be back here beginning it again at 2 o'clock.
Lunch will be available for.
Oh, and for presenters? And lunch will be available for presenters in this room. I'll put up our, is this someone's presentation?
No.
That's typically moved presentations. There's a test from her laptop. We can put it on
that
on Tech Savvy one.
Yep.
And I did also email her the voice thread. I don't know if you want to display, like, just have it run, or just tell people to check the website.
We have it written in here, right?
Yeah, that is that.
Then, yeah, we may as well, no, it's not listed here.
Or that.
Is it that
Okay,
one?
I thought it was out.
Yeah.
Yeah, we may as well just run it.
Okay, yeah. Someone else has dropped out. Yeah. Theoretically, I've never downloaded a voice thread, but I assume I can and put it on actually.
Was this one or this one? I think it was this one. Was this the